So congratulations, you've reached the last chapter in the book, Nanobiology. In this module we're going to give an introduction to some terms, and in the next outline some basic molecular biology. We're going to discuss enzymes as nanomachines, molecular motors, and then end up with a discussion of fluctuations both in gene expression and gene splicing. Biology is the ultimate example of a system that is completely dominated by fluctuations. This was recognized by Charles Darwin in the 1840s when he produced the basis of his monumental work on evolution by natural selection. Darwin noticed that there were random variations in the way that a species appeared. Biologists call this a phenotype. But the environment would act to put selective pressure on particular members of the species and evolve them to the fittest type. As the result of the discovery of the structure of DNA in 1953, we now know that one source of these random variations in phenotype is random variations in the genetic code itself, which then gets translated into random variations in phenotype, but in a very complex way that is still the key research topic of modern biology. So let's look at how living organisms are made up. We're going to talk in general about eukaryotes, which is you and me, all the multi-celled organisms. These have a cell nucleus that packages the DNA. In our cell nuclei, there are approximately 2 meters of DNA in total, several billion base pairs of DNA, packaged into a small compartment consisting of a lipid bilayer, which is the cell nucleus. Outside of the cell nucleus is the cytoplasm, in which sit all sorts of machines necessary for the function of the cell. And finally, the whole thing, shown here as being about 10 microns in diameter, is encased in another lipid bilayer. Prokaryotes have no nucleus. These are, for example, single-celled organisms like bacteria, and they simply have all the genes they need essentially turned on all the time or regulated in very simple ways. In the eukaryotes, the genes can be regulated in very complex ways, giving rise to different patterns of gene expression in different cells, and therefore the cell specialization that's needed in a multi-celled animal. Some of the components visible here, in addition to the membranes we just talked about, are the ribosomes, which are the factories that make protein from RNA code, and the mitochondria that we talked about in Chapter 6. I'll just mention there's a whole other class of living creature, the archaea, which are predecessors of the eukaryotes, and in their own right extremely interesting. So what are proteins? We know about DNA. Proteins are the things that most of the machines in the cell are made of, although many machines, like the ribosome that we just discussed, have a very high content of RNA as well. These are some examples of protein structures formed by downloading data from the structure database at the National Institutes of Health and using a model program to show what the crystal structure looks like. This is a transcription factor, a protein that controls gene expression, bound with a piece of DNA. So here you can see the double helix, and here the transcription factor sitting at its binding site. This next protein here is a kinase. That's a protein involved in signaling pathways. Its specific job is to attach a phosphate group at a particular location on a protein to mark it for a certain destination or pathway in the cell. You see this little hole here through which the chain of the protein could pass. Finally, this is actin, a scaffolding protein. This makes the filaments that give the cell a structural strength. So this is like the 2x4s of which the cell is built. These are all drawn on different scales. Each of the little dots here represents a single atom. So you can see how the length scale has changed as we've gone from this small protein, this intermediate protein, and here a rather large protein. Looking at protein structure at a basic chemical level, proteins are made by stitching together amino acids. And here is an example of an amino acid. This is the backbone of the amino acid here, terminated here in a carboxylate residue and here in an amine residue. The R stands for one of 20 chemical groups, and we'll talk about those in just a second. These amino acids are stitched together by chemical reactions in which this C-terminal end reacts with an N-terminal end of a second amino acid, expelling water. So we go C, N, H, C, and you see we've gotten rid of an oxygen and 2 hydrogens. The exact properties of the protein depend on the pendant groups attached to this peptide backbone, as it's called. I show you here some of the side groups that look like just chains of hydrocarbons or an organic benzene ring. These are all residues that are hydrophobic. So regions of a peptide chain rich in these residues tend to push to the inside of a chain and guide its folding. Hydrophilic subgroups, on the other hand, have components that are charged at neutral pH. This is important, and we'll return to this later. This means that they like to be dissolved in water, so these residues like to sit on the outside of a protein. And it is these factors that drive the folding of a protein. There are a number of in-between groups shown down here, and these can change their character depending on the local pH, and therefore switch the function of the protein. Finally, I come to the genetic code. This is the fundamental relationship between the code listed on the DNA, transferred to messenger RNA, about which we will talk in a minute, and then converted by the ribosome into a specific residue. U here stands for uracil, which is the RNA analog of thymine. So you see a UUU on the RNA, which would correspond to its complement, an AAA on DNA, gets converted into phenylalanine, for example. Over here, GGU, which is on the RNA, equivalent to a CCA on the DNA, gets converted into glycine. Notice how the code is redundant, with sometimes as many as 4 different so-called codons, groups of 3 bases for each amino acid residue, and sometimes fewer. For example, here there are 2. Finally, there are control signals. You notice that UAA, UAG, UGA are stop. And then poor old tryptophan here has just 1 codon.